Good evening to this evening's ReGen Ag Chat. There we go. I was going to say webinar and I got confused. So this is number 16, and this evening we're joined by Ian and a special last minute guest, which is Tanja from the Netherlands. So we'll come on to them in a second. So Nick, what have you been up to since our last one?
Well, the kids have run away with the hairdryer, so I am looking particularly bedraggled. But we did the last one exactly 4 weeks ago today, and we did just mention Ukraine. And then 3 weeks ago, Reno got out of bed one morning and looked out the window and saw the stock trailer and said, and we were listening to all the Ukraine stuff, and said, I'm gonna take the stock trailer full of stuff and go there, which started this kind of snowball. And 3 weeks later, we've sent 4 loads to the Ukraine border, raised £20,000 on a GoFundMe site. This is all us and a few good mates. And we've had donations of medical stuff. So altogether, I think we've sent £22,000 worth of medical stuff and loads of food and some wheelchairs. And we've just, it's been completely knackering, if I'm honest. But we've met some amazing people. We helped some refugees across the border. When Reno went, they picked this, it just kind of brings everything home to you. They brought this lady back, 3 families, but one family was a lady. She was like a professional. She worked for a medical company, and she came over to Amsterdam with her son, who was about 9, and her daughter-in-law, and she'd left her husband, an older son, over there to fight. And she said to Reno, she said that she kind of previously thought of herself as like a very confident and with it lady, and she just said, I'm an absolute wreck, and I just, my life's gone to tatters in 4 weeks. And I think so we've just been on that full on for the last 3 weeks, and I'm now a bit knackered, but yeah. And then carbon calling wise, it's tootling along. Speakers I'm trying to find beds for speakers if anyone wants to give, who's a local farmer who wants a bed.
That's quite dubious, or could be slightly dubious.
I know,
ideally a separate bedroom.
At the minute, I don't know if Tim Nicholson's on, but I've got in his house there is Michael Blanche, Alec Brewster, and who else? And Janet Hughes, and maybe Patrick Holden. So that will be maybe the party house. But yeah. So it's all going well. I'm just tired.
Well,
how's lambing? How's lambing?
We're nearly through it. So I'm tired for a different reason, for getting up in the middle of the night. And I've started to mount my own biltong. Both of them seem quite, what's the word? Small compared to Ukraine story, but my new biltong box is very exciting.
Biltong.
So it's dried South African dried meats. So I'm drying my own meats.
Look at you.
No, I've become very anyway. It's what lambing does. Sleep deprivation makes all things happen. So
I know. Well, yeah, I know. It's just quite hard without sleep. But yeah, but I'm looking forward to this. I haven't. You've done most of the kind of legwork with this, so I might sit relatively quietly and just pop in when I need to. But yeah, I'm looking forward to it.
So this evening we're going to be joined by Ian Bell and also Tanja Dekker. So, and the main thrust of the conversation really is about food webs within soil. So looking really at soil biology and understanding that a bit more. The key reason that Ian's invited, obviously other than he is very knowledgeable, is that he's planning to do, or we're doing a workshop with him at Carbon Calling in June. So he's coming along to Carbon Calling along with his microscope, and we'll do a bit more work with him out on the day, either on the Saturday or the Sunday. We haven't quite decided that yet, but anyway, you will be in Cumbria in June. So I don't know whether Ian, if you're happy to, just to give yourself a quick introduction before we make a start, and then Tanja, I'll ask the same.
Okay, cool. Am I live now?
Yep.
Well, hi everybody, and thanks Liz and Nick for inviting me along to do this, and also along to Carbon Calling, which I'm really looking forward to, and hopefully it'll be quite a fun thing. We'll bring our microscope along and a big TV, and all being well, with no technical glitches, we'll get them all hooked up together. And if any of you want to bring some soil along or some compost along, we can get it up on the big screen and have a quick scan round and see what's there or isn't there. So a little bit of my background, that I guess going back a bit, was family farm in the Dales, dairy cows, sheep, Harper Adams degree, and then later an MSc, all in conventional ag, worked for Velcourt, and sort of pushing all the buttons on things that we maybe know now probably shouldn't be, trying to get as many litres as we could and many tonnes per hectare, and a lot of damaging practices. My career was sort of cut slightly, hands-on farming career was slightly cut short with an accident and a spinal injury, but looking back now, it's maybe a blessing. It stopped me doing any more damage to soils. Following that, did work, bit of advisory work for 10 years or so with a consultant from Ireland, working on EU type projects in different third countries, and diversified a little bit in sort of what started as a business selling lamb online from the farm here at home, then fledged into an outside catering company, and did that for a number of years. But for me, that step away, even though I was living still on the farm and family farming all around me, was probably quite important, because it allowed me to sort of take a step back and look and think about what I was doing before. And that led into sort of coming across the likes of Alan Savory and Gabe Brown and Joel Salatin, and the route probably a lot of you will have gone down, and becoming thoroughly absorbed in reading as much as I could, books by Charles Massey and the whole plethora of information that's out there. Then I came across Dr. Elaine Ingham and the work she was doing with the Soil Food Web School, and I'd done a little bit of training on the grazing with holistic management, but was still sort of scratching my head how I could take what was an interest and a passion into something that was a little bit more than that. The training with the Soil Food Web School kind of opened that door for me. And so for the last 2 and a bit years, it's been a lot of studying, a lot of research and learning, and I think that learning will continue for a long time to come, because there's so little we know about what's happening in the soil. So we've now qualified to do as a soil food web lab, so we can do analysis and provide you reports telling you what's in the soil, whether that's your FTB ratios or what biodiversity and levels of the different microbes you have. So that using that as a tool, we're also in the progress of hopefully this year completing the consultancy accreditation program with the Soil Food Web, and Tanya's on the call with us. Tanya's a really experienced Soil Food Web consultant, and the two of us have a weekly meeting and chat about ideas, and Tanya's sort of been for the last 18 months sort of guiding through the ups and downs of making compost and trying to get really good compost with a lot of diversity. So we're looking for that fungal content, but we're also looking for diversity of protozoa, bacteria and nematodes. And I think gradually, after sort of a lot of sort of early fails, we are starting to get piles that have got that quantity of fungi in that we want, and then now starting to sort of take that out onto farm, and working with, at the moment, sort of 4 or 5 farmers in the Yorkshire Dales and across in Cumbria this year, varying from sort of 6 hectares up to sort of, I think, maybe 50 hectares on some of the places, but certainly not taking whole farms on, but looking how we can what we can do to move those soils on. And yeah, so that's where I am.
Thank you very much. Tanja, welcome.
Yes.
You had about 2 hours notice, so thank you very much for joining us.
I'm gladly do it. My name is Tanja Dekker. I'm stationed in the Netherlands. I'm a soil food web consultant, graduated in 2019. Once I started my own practice, Dr. Elaine hired me as a mentor. I have 60 students worldwide. So I start my day in Australia, go up to Thailand, go to Cambodia, then do all of Africa and Europe, and then I do New Jersey, and then go cross over to America. That's my day. That's what my day looks like. World travel every day. I also have 5 individual projects myself. So I have worm hotels in my own town because I want neighborhoods to spruce up their own neighborhoods, right? So we've been doing that for a couple of years now, and all of their gardens are really wonderful. So now they've branched out to take care of the trees that are in their street, which I find wonderfully. And actually they produce so much compost that I get to use it in some other projects that I do. For instance, a food forest that we've just put in this month of March, and I help dairy farmers switch over to regenerative agriculture. That's me.
That is an incredibly varied introduction. Thank you. I like the idea of a worm hotel. So is it that you just briefly talk to me, what is a worm hotel?
Yeah, so I have worm hotels that are above ground and ones that are below ground, right? Sometimes you have these deposits that are below ground. You open up a lid and then you put in your garbage, right? So I turned one of those around into a worm bin. And that means that my municipality, my city, does not have to get that for less than 18 months. So we don't have to send out a driver, a pickup truck, like a huge truck. It used to have to go and pick it up 70 times a year, right? But now they don't have to do that because they have to do it once every 18 months. So that is a huge cost saver because now they don't have to collect the greens because it's being produced into compost and then it can be applied immediately.
So this is your food waste. Yeah.
Food waste, yeah, from regular people in a regular neighborhood, right? Now, if the rules are if you are above ground with your hotel, then you can't put any processed foods in there because it would attract rats. So they can only put in, if you have cauliflower, you eat the cauliflower, but the rest of it you put to the worms. So the uncooked vegetables, right? That's what you put to the worms. But the other rule is that you have to put in twice as much brown, like carton or paper or brown paper or anything woody. Then you put in green so that we get a nice high fungal count. And that's really going to improve their lawns and the trees and the flowers and whatever they have. So
how many people are in this system? Or how many in the neighborhood? Or
yeah. So it's the neighborhood has to apply themselves. 5 of them have to say, I'm going to feed the worms. And then up to 25 other neighbors can sign up afterwards, right? And they get a little pass. So they open up the gate, right? And because other people could vandalize it or put in plastic or not know what they're doing. So it's regulated. I'm working with my city to do that. And they're very happy with it, right? Because it saves a lot of money and you get all of this compost and you can apply it to your soil.
Oh, that's amazing.
Yeah, but the ones below ground, you can put in anything, mayonnaise, your rest of your dinner, anything you can put in. It's just that the rats can't get to it because it's completely closed off, right? But the ones above the ground, rats can always get in because they're really very athletic, I guess.
Very logistic question, or how do you get it out, the below ground one? How do you get the compost out?
Yeah, well, that's regulated already. We have these bins. One is for paper, one is for glass, one is for rest, and then one is for green rest stuff. And then they just have a machine that picks it out and just dumps it into a truck. And that's what it was anyway, but now they have to have an empty truck to empty out the worm compost. And then we collect the worms, put them back in a hotel and lower it for another 18 months.
The Netherlands is just so much better than us. Anyway, before we get into we're moving to continental Europe. Thank you very much, Tanja. There's a few questions coming on worm hotels, so we'll come back to them.
Sure.
But Ian, I didn't know whether you just wanted to flick that first slide up just as a bit of a sort of intro to what we're on to talk about.
Yeah. That one there. Oh, can everybody see that?
Yeah, just needs to be flicked onto full screen.
Is that, have we got the
Next one. There we go. Perfect.
That's cool. Yep.
Yeah.
So yeah, there's a lot going on here, but and some of you will have probably seen this before, and I won't go into too much detail, but if we sort of quickly work through it as a flow, the bit about the plants, I'm sure we all know that plants are our solar panels. They're the thing pulling the energy down into the soil, pulling carbon down into the soil, and choose the mouse, please. And converting the carbon into sugars, and they can be putting up to 40% of the sugars they produce into the soil, and that isn't purely because they've got an excess of sugar. It's because they know that they can use it as a trade-off. And those sugars will be lots of different recipes that will be calling for different nutrients from the bacteria and the fungi. And the bacteria and fungi have the capability of solubilizing nutrients from sand, silt, clay, just down at the bottom, which, so when we conventionally look at a soil chemical analysis, we're only measuring soluble nutrients, but there is a wealth, any amount of nutrients locked up in these particles. And then the thing that is missing is these microbes that can solubilize that. And from that point on, we've got two different sort of methods of the plant being fed. The sort of conventional nutrient cycling is where we've got bacteria and fungi, and then the protozoa will come along and it will eat the bacteria. And as our American friends say, it's start of the poop loop, because the protozoa doesn't need a lot of those nutrients, and they're passing out those nutrients right next to the plant. So it's getting all the nutrients it needs from this cycle. And then protozoa might be eaten by nematodes. Nematodes might eat nematodes. Protozoa might eat nematodes. And we're just suddenly building up on this layer and layer of nutrient cycling. And it's all happening in the rhizosphere, in the sheath around our roots where these sugars are. So plants are getting the nutrients they need, the water they need, plant defense chemicals, hormones, everything a plant needs to grow is capable of being provided from this soil. But the thing that is missing is particularly the fungi and the protozoa and the nematodes. And as we move up, the arthropods, the dung beetles, the worms, the things that we can see when we take a spade and have a look in soil. So those are, that idea of our spade being our most important tool. Let's see what's there with the bigger organisms. But what we can do with the microscope is check out, and we can quantify and measure all these smaller guys. And this idea of nutrient cycling has been around for a little while. Dr. Elaine Ingham was one of the first researchers to sort of hit on it and research it. And then latterly, Dr. James White has been coming up with his theory of rhizophagy, where plants are capable of actually absorbing a bacteria and a microbe, stripping the nutrients off it needs inside it, and then spitting that bacteria back out, but in a form that the bacteria is still alive. So just an amazing amount of symbiosis and interactions going on and signaling between plants and microbes. And we know nothing, we know 1-2% of maybe what's happening under there. But that's the challenge for us to learn more. But at basic level, there's that understanding that there is enough nutrients within the soil, and 78% of our atmosphere is nitrogen, and there's we all know about the rhizobia that can form nodules on legumes, the clover, but there's a whole wealth of other free-living nitrogen fixers that don't need the symbiosis with clover. So what we need to do is re-establish these guys, and then re-establish them in a balance. So if we just click on a couple of slides, go on to the next one and the next one. So this one here, I'm not going to the full detail of it, but left-hand side where we've got dirt, bare ground, we've had an earthquake, or we've plowed and tilled and plowed and tilled and just created sort of ground zero of soil. We've virtually got no fungi. It's completely bacteria dominant. And if we leave this for nature, it's going to take a few hundred years, but she'll send in those early colonizer weeds, those with little short roots, grow fast, loads of seeds. She wants that ground covered, and gradually we'll start to get a little bit more fungi. Then our brassicas will follow that. So, that would be commercially an oilseed rape or any of the kales and other things there, early succession. And then we're moving on, we're gradually getting more fungal dominance. So we get our early succession grasses and different vegetables. And this is, I guess for a lot of people listening, this is where we want to be. This is what we're talking about. We're talking about productive grasses, and all our cereals are, the members of the grass family. And we want soils that are an equal balance of fungi to bacteria, or just slightly less. So in that range 1 to 1 or 0.75 to 1. And if we let nature just take her course, it's going to take a long time, but she will keep moving on. We will start to get woody shrubby species coming in. We'll get deciduous trees coming in, and finally, a few hundred years down the road, we'll end up with old growth forest. And that soil could be anywhere between 100 to 1,000 parts fungi to 1 part bacteria. So completely fungal dominant. And then the key message in this is where we should be in the middle with productive grasses. We go in and we overstock, or we put a lot of fert on, or we till, or do all these damaging practices, and we're just pushing it back in succession all the time, to a point where the soil is in a great place to grow weeds. And therefore we come in and we plow the weeds, we put more herbicides on, and we're just trapped continually pushing it back in succession, and more and more on. And as the saying going, we maybe become more on farmers. It's of utmost importance, and highlighted at the moment, is the cost of doing that is that's machinery costs, that's chemical costs, that's fertilizer costs. And we don't need them if we can just get this balance in the soil. And it isn't just about the fungi and bacteria, it's about, as we move across that line, increasing the amount of diversity of microbes. So that's the protozoa, the nematodes. We want a bacteria feeding nematode, we want a fungal feeding nematode, we want the nematodes that predate on other nematodes, and within that there'll be the root feeding nematodes, which there's something like 95% of the research done on nematodes focused on the 5% of nematodes that are the root feeders, the plant pathogenic ones, and very little research done on all these other guys. And the point of what we're trying to do is get that equilibrium, that balance back in there. And the root feeders can't move very fast, so the predators, that's a nice easy prey for them. And if we get the predators there, they'll keep the root feeders in balance. And as with all things in nature, nature is more than capable. It's had 3.8 billion years to work out what these balances are we need. So that's the background of what we're sort of looking at, and the theories behind it.
And I suppose a question for me, and Tanya may also, June, I'd want to add to this, which is the challenges with researching this is, I'm guessing, how do you do it without destroying the bit that you're trying to research, in terms of understanding those interconnections of what's happening in that root zone? How is it done, I suppose, is the question. It's a badly worded question.
Do you want to go with that, Tanya? Or...
You're right on track Ian, you go take it.
Okay, so in terms of researching it, we've got this tool sat right next to me, the microscope, and so I've got some trial plots that we established early last year, and it's we're also introducing the idea of grazing management in there. We've got they're all contiguous plots, but they've got a fence line across. One field's set stocked, and the others in ReGen grazing, mob grazing management, and each field has a control and a treated, so we can measure. We're measuring grass growth, we're measuring worm counts, we're measuring water infiltration, we're going to measure carbon over time, we're measuring changes in the organic matter. But importantly, I guess in relation to what we're talking about tonight, we had baselines of what soil microbes were there, and we're sampling and measuring that change. And the end goal of that is to, that soil had no fungi, it had maybe an odd root feeding nematode, very little protozoa, it was all bacteria. And that's land that has just been set stocked. It's had a little bit of spot sprayed herbicide, but it's been permanent pasture for a very long time. So in terms of what we consider the things that do a lot of damage to the soil, the ploughing and the chemicals that go with arable farming, it's not had that, but the damage that the set stocking has done, where we've got roots that are a few centimeters long, it's just, there's nothing getting down into the soil to push quality exudates down. And grazing management's an important part of that, but we've also looking at applying soil biology, and this year we're going to focus a little bit more on the humics, the fulvics, the fish, the seaweeds, and the other sort of stimulants that we can get in there. So that's how we would measure it. Sorry, long way.
No. So it's basically you're looking for shifts in population numbers, aren't you? I'm sort of thinking you're actually trying to understand what's happening at the root. There's an element of understanding what's happening with a population shift.
Yeah.
So
absolutely.
before and after, sort of all tracking that.
And it's getting away from this reductionist science of doing it in a petri dish in a lab, where you just can't do it with soil biology, because if you've only got one agar source, then that will only feed one type of fungi. It will only feed certain types of different microbes, and we just can't measure that. But it's more of a holistic sort of view of looking at this. We're measuring, fortunately the national parks have been really great, and they're helping with measuring different plant species and how they're changing. We've got a plate meter so we can measure grass, and it's trying to pull in as many parameters as possible, and we may never know how they all interact, but hopefully it's I think Nicole Masters quite often says, if we don't measure, we can't manage. So we're measuring as much as we can on that, and the key to a lot of that will be the trial plots from my sister and brother-in-law's farm. Is when they walk across that paddock, they're going to get that feel, that intrinsic sort of knowledge of is it changing, how does it look, are the cows preferring to eat one plot than the other. And when these things are sort of, those are just 10 by 10 meter plots, but when we sort of upscale it to what we're doing this year, we're going to get a lot better idea of stock performance, and hopefully we'll see health benefits. And it'd be really nice to tie into the work that Dan Kittredge is doing with the beef nutrient scheme, and one of the farms which is going to be beef grazing. We're looking into the logistics of sort of monitoring that, getting a steak sent out to one of Dan's labs, and contributing to what they're learning
from a nutrient density, so the meat perspective.
Yeah, so if you haven't come across Dan Kittredge and the Bionutrient Association, brilliant guy, amazing guy, and does a lot of sort of regular webinars and then presents them really well. He's worth looking up on YouTube. And for the last 10 or 12 years, they've had a plant nutrient density program going, where they've looked at something like 12 or so, I think, different plants, whether that's wheat or a courgette or a tomato, and got thousands and thousands and thousands of data points and measuring how nutrient density change and linking it hopefully to farming methods. So the beef one, he admits himself, is going to be vastly complicated, but if we're going to make these claims about where we're producing nutrient dense food, these type of projects are so important. We need to put our money where our mouth is and have these measurements. And it's quite exciting. And I think similarly with carbon, that methods of managing carbon at the moment aren't great. There's a degree of error with it, but we go with what we've got. And I think, am I right, Tanya, that it's Dr. Ingham saying that the is it near infrared spectrometry or near infrared is probably going to be the best way of managing, but it's 4 or 5 years away from actually being able to be used. But that will have the ability to measure the carbon in fungi. Is that correct, Tanya?
That's totally correct. And I know that they're developing this. So I'm hoping it's this year or next year when it comes out.
Cool.
Yeah.
Nice. Oh, that's a bit quicker than I thought. Yeah.
So
can I make a point about
please do.
One thing that people really always want to know, and that is, how can we get rid of weeds? Weeds is another word for pioneer plants, really. But as you look, it's way on the left side, right? It's on the side with bacteria, and it's because they respirate the nitrate, right? As if you add more fungi, they respirate ammonium, and that suppresses the germination of weeds, or what we call
down a bit.
Pioneer plants. And when they do pop up in your productive grasslands, then really look at them as indicators, because they're telling you something that you're lacking, and that's why they exist. So that was my only point.
That's a really good one as an indicator. And that we see, conventionally we see a weed as a problem, but something like a dock may well be an indicator of surface crusting, of compaction. That's nature's way of sending something with a great big massive taproot to break that compaction and pull up minerals from lower down. And let's change our mindset and think of the reason, the weed is a symptom, but what's the problem behind it?
And so if as a lot of us would have or feel, so we're in that cycle where we moved from bare soil, but we're in sort of weedy type areas. We're not quite in productive grassland. So where do we start in terms of understanding our bacteria fungi ratio?
Cool. So I guess the starting point would be taking the sample. Now, you can get a microscope yourself, and we'll come on to looking at a slide here, and you can pick them up fairly cheaply and have a whiz round and see what's in there, or you can send a sample away to a lab like ourselves, or Daniel Takiri or Eddie Bailey. There's a few in the country now who will give you a real quantitative analysis of what's in there. So that's your baseline, and then monitoring it as it goes along. Do you want to jump onto the
let's have a look at
that point? So what we've got here is from, we've got 4 different slides. So this one is from a compost pile of ours, and we'll just sort of point out some of the different things we're seeing in here. So first thing that I notice is we've got these sort of, can you use the mouse please a little bit? These big brown aggregates. So the darker browns are humic colors, the honey colors are more the fulvics, and we know we've got the organic matter there as a starting point. What else we're seeing? If we just move up a little bit on this, so this is, we're on 400 times total magnification. So let's just stop there. So just on the left there, if Olivia just points that out, see that guy there? So that's a testate amoeba. And they're potentially going to eat something like 10,000 bacteria a day. And they don't need very much of the nutrients that's locked up in that bacteria. So if that's right in our root, next to the root, most of the nutrients it's consuming are being passed out and there as plant available food. There's another test date, I think, a little bit lower down. So just tweak the focus, please. Is that another test date?
It is.
Yeah, in the middle. And then, so the thing like a rugby ball, that's a fungal spore.
Yeah.
We're not certain when that would germinate, but one would hope when this compost goes out that if the moisture's right and the conditions are right, that's going to germinate into fungal hyphae. If we just keep moving on. And right, here we go. So this is what I start to get excited about with compost. So these brown strands, so if Olivia just points these out, these are fungal hyphae. And this is what we're not seeing in soil. And we know this is likely to be a really good beneficial fungi because it's an even width. It's got the little, let's go into that one, that's even better. So let's just tweak the focus on that. And if one of the sort of tells is a little scepter, the walls across, so if Olivia just points that out, we know we've got an even width all the way along and it's really dark brown color. And yeah, if we can start and get these in our soil, then we know we're on the right route. And the great thing with it, the fungi, as they get older, the walls are made of chitin, so that young fungi might have a C to N of 20:1, but as they get older and those cell walls get thicker, that's the point they're laying carbon down in a really stable form. Like this won't be a mycorrhizae fungi, that will only grow where there's an interaction with roots. But if we saw this from soil, it may well be a mycorrhizae. And then those are you may have heard of the term glomalin. So where it's a glycoprotein that is so long lasting in soils, and that's the point we can really pull carbon down. But yeah, when we see these things, Tanya, we get excited, don't we, in compost?
Yeah, this is really great to have this really strong fungal hyphae there. That's really good. Yeah.
And then sadly, if we just swap a slide over, Tanya, do you want to just talk for a moment or two while we change slides over here and look at the soil to sort of compare the different?
Tanya, can I just ask? I mean, this is, for me, this is absolutely amazing. And yeah, I just love it. It's quite addictive. But is this one of when you're trying to get farmers are just kind of starting their journey into this, is this the kind of good starter drug just to get them kind of begin to get hooked?
It is totally, and the compost is really an inoculation. And then once the biology has found again the relationship, a symbiotic relationship with the plant, which is the whole thing. When you are a plant, normally you would make all sorts of exudates, which is really a batter. The sun shines, right? They take the CO2 out of the air. They break off the O2, oxygen they don't need, right? They throw it away. They end up with a lot of C's, carbon chains. They make sugar out of it, loads of sugar. And they make proteins out of it, loads of protein and carbohydrates. Now, if I send you to the kitchen and ask you to put in a bowl a lot of sugar and a little bit of flour, as for carbohydrates, and a little bit of protein, maybe an egg, and I make you batter this, you'll make a batter, right? That's what plants do all the time. And the sugar is a code for what they want. So the sugar has a coding, and they want calcium or magnesium or borium or manganese or whatever. And that's how they start the exchange. Now, if you are a plant in a chemical system, you're given this in a water-soluble thing. So you're going to just take it up as a diffusion, right? The concentration is higher outside of the roots than inside the roots. So it just seeps in. At that point in time, it will stop putting out exudates because it's getting nutrients without having to do any work.
Yeah.
It's not that the plant is lazy, it's that it's now addicted to the drug, to the easy minerals that are water soluble. So if you're going to switch over, you need to teach your plants to do the work again. Set out, push out the exudates, make these symbiotic relationships, and then you'll have a really good kickoff.
And as far as kind of the getting away from the drug addiction, because there'll be maybe people on the call who are still using, I guess a lot of people on the call will still be relatively conventional.
Totally understandable.
And how long can, because when we stopped using fertilizers, we had a dip for maybe 2 years, 2.5 years, and then kind of came out of it. Is that typical?
Yeah, if it's with me, I see if a land has been fertilized for 30 years plus, I need a remediation season. I need to turn it around, because especially if glyphosate has been used to terminate certain crops, it changes the organic matter, and I have to do something in order to fix that. So we put in an understory cover crop, right, for 1 season, and that's from the fall until the springtime. And we hope to see a huge improvement just with the cover crop. And then we do terminate it by a roll and crimp, and we plant the cash crop immediately as we terminate the cover crop. And in between the rows, the cover crop can come up again, but the cash crop has to be higher. And if we can do that, then we're successful.
Okay, thank you.
I suppose, Nick, also some of that is we don't, or that could that be speeded up by compost application? I suppose with that 2.5 year is based on natural inoculation or changes in your soil compared to putting on additional amendments. I'm making it up, but that's what I'm just thinking.
You're totally right. What we can grow in compost is limited, right? We can't grow as much microbes as we want, but a plant can, because it has all of the ingredients that a microbe wants. It's the nectar of the gods for them. They can't get it anywhere else than from a plant. So a plant can grow its microbiome a lot quicker than we can, but then it needs to be weaned off the fertilizers, right? If it has a full-on relationship, symbiotic relationship with the microbes, it gets all of its nutrition from the soil and from the microbes. And if there is, in the transition, we usually do reduce the fertilizer until the plant has now gotten used to doing both, and then we can take away the fertilizer.
I think what we do have with the biology is that ability to kickstart it.
Yeah, that's it.
And we're not, this is an inoculum. We're not trying to put all the microbes on that soil needs. We're just putting hopefully the mix of fungi, protozoa, bacteria, and nematodes. And the real difference is you put fertilizer on and it diminishes. It's only going to get less until you put more on next year. If we put microbes in, then your protozoa, they're reproducing every 8 hours, your bacteria every 20 minutes, fungi. What's the range with fungi, Tanja, in terms of reproduction? Is it a few days?
No, actually, it could grow every hour if conditions are really well. Then it can grow really fast. But of course, it grows fastest if it gets the better from a plant.
And so it's only going to get more. And this is kind of where I think the interesting bit comes in is combining different techniques. So the land we're working with is all grassland, and if we can move away from set stocking and introduce proper grazing management, mob grazing, holistic grazing, regen grazing, whatever you want to call it, but where we're allowing those plants to get a proper rest, and we're not overgrazing, and we're not taking them too short, that they've always got those solar panels and they're not using that root energy, so the roots can start to go down and they can start to push more sugars out for the microbes. That a lot of soils, these microbes will be there, hidden dormant, and particularly in the area where we are sort of I guess there'll be people on the call from all over, but the Yorkshire Dales area, the Eden Valley up to where you are, Nick, that we haven't had that history, that legacy of a lot of tillage and chemicals. And organic matters are certainly on the farms I'm seeing that, you know, they're reasonable. I've not seen anything below that 3% threshold, and it's below that there is, you know, if you've got a soil and the organic matter is less than 3%, there is no point putting biology out, because that's the threshold below which it can't survive. But a lot of these farms, you know, the soil is damaged, but I would hope we can, you know, by combining all the tools in our toolbox, that we can move it on quicker than something that's in East Anglia that's had,
near me.
Tanja said, a 30-year legacy of lots of chemicals, very little organic matter. So, you know, time will tell with this, and it will vary farm to farm.
There's just a couple of questions on that. One, which is, someone's got docks and they don't really like the docks. How do they get rid of the docks? But I'm guessing it's a process. We have to accept them. And this idea of actually how we'll come on to, which is how do you start building that fungi within the soils, which
Yeah,
you've sort of started talking about.
So with docks, get a spade, go and dig a hole, and, you know, is that dock indicating a hard surface compaction, or have you got compaction further down? And address the compaction. It may well be a calcium issue, but sort of, if we look at addressing the problem rather than the symptom, that would maybe be first and foremost. But personally, you know, I'd be loath to sort of go down the route of putting a chemical on there, because we're not addressing the compaction problem, and if anything, we're going to set the stage for an early succession soil. And it's for those with bricks meters that it's really interesting, you know, where you've got weeds. We've done it on the farm here, where there's a patch of thistles, and the bricks levels in the thistles was way over twice that in the surrounding grass. And the interesting thing with that, there was, the field had a mob of mule gimmer lambs in there, and there was 4 or 5 of these mule gimmer lambs were constantly nibbling away at these thistles. The rest wouldn't touch them. And when I had a chat with my uncle, and it was the same lambs coming back, they'd been pet lambs, and there was no learned behavior from mother as to what it should eat and what it shouldn't. They were up for trying anything, and once they tried these thistles, they were hammering them. So we, you know, there's a lady, I can't think of her name, in either North America or Canada, who's sort of done a lot of work about training livestock to eat weeds. And, you know, when we realize there's a lot of nutrients in them, then, you know, if we can use livestock as a tool and grazing management. I know docks can be bitter, but I've heard of people, you know, when that mob moves into that paddock spraying molasses or something on, just to try and get them into the habit of eating it. So grazing management, and, you know, that might be in terms of the compaction. We look at pulling, you know, this idea of drag and drip. So we pull a subsoil through with biology going down behind the legs to get biology into the soil and help break that compaction up. But that dock is an indicator, and, you know, where we attack it with sprays, we're kind of setting the scene for another problem to come along.
Just another question, which is in terms of if fields are sort of shut up for hay rather than silage, do you mean, so they might be graze short for some of the year and then obviously allowed to go long for some of the year? How does that affect in terms of rooting structure? And is that is it a compromise, or is it still not going to be as good as a full mob grazing system?
Absolutely, but I think it's like, as long as we are aware that we're doing a little bit of damage, maybe that sets stocking at lambing time, and inevitably we end up taking those covers down lower than we want, or we come through and cut. The real key to it is realizing it's going to take slightly longer for that plant to recover, and we allow for that. And where the damage happens is when the regrazing happens too soon. So if we'd set stocked at lambing time and then 2 weeks later we came back round and we're continually hammering that regrowth, then we're going to be reducing the roots and causing problems. So it's this idea of holistic management is you have to do things that aren't optimal, but being aware of that and just allowing something in your system for a little bit more regrowth. Like the theories of soil health and the practicalities of farming have got to work together.
And so I suppose the key point really is that we can put as many composts on as we want, but if we're not changing how we manage the plants, then it won't have an impact. It will have a short-term impact, but actually it's just kick-starting that system, and what we really want is healthy plant populations driving it.
Exactly. That it's the management has to coincide with the treatment, and that I think you get the most bang for your buck with compost is if you can get it in furrow. So and one of the farms that we're going to be 3 of the farms actually we're going to be working with this year, they're all either introducing a herbal ley into a field that's had a winter cover crop, or looking at we're going to try and get it introduced into a cover that's already there, into a pasture that's already there, without burning off or ploughing. And that there's this idea that when a lamb's born, it as it passes out into the world, it gets inoculum from its mother of the bacteria and the microbes it needs for a healthy immune system. And whether that's a lamb or human, it's nature's way of setting something away with a starter pack of microbes that will get it through those dangerous early days. And we can do the same with our seeds, that each seed is already equipped with all the sort of cofactor enzymes needed for its growth. And if we can coat it, there's various ways of looking at this. So Dr. David Johnson, the guy behind Johnson Sue, he's sort of making really good quality extracts and then using a cement mixer and putting I think he's only putting like a pound or 2 of a really good quality extract into a cement mixer with his seed, turning it around, putting it out onto a sheet, letting it dry. And the microbes are going to go dormant, but once they go into that seed bed and there's moisture and things around, then they'll come back to life, and that germinating seed will be given its best start in life. The method we're going to be looking at is we've developed a sprayer based on the guy Steve Erickson in New Zealand, Chaos Springs. If you've not come across him, look Steve up. He's brilliant, quite active on social media, and I think he trained with Dr. Elaine 20 odd years ago, and he tried to adapt sprayers, conventional sprayers, but just found he was sieving out so much biology to get it through the jets and filters that he nearly defeated the object. So the spray he's made has really wide nozzles, and in effect you're spraying slurry out. So the analogy I've been using, and proof will be in the pudding, it's just been finished, and we can maybe show some videos of this, is that the conventional way of making an extract is we're sending a microbe on a Bear Grylls survival course. It's going out there, it's got nothing on its back, it's got no food, it's got to find shelter pretty quickly, it's got the sun beating down on it, and everything's stacked against it. Whereas I'd like to think that our microbes are going to be going glamping, that they've got a cozy house with them, they've got food, they've got that organic matter around them, and then the theory is they'll have more chance of surviving. But as I say, like Steve's been doing this 20 years, so he wouldn't have spent all that time developing his sprayer if it didn't work. But you've done quite a lot of work, Tanya, with on a smaller scale using compost slurries, haven't you, and found a real benefit?
Yeah, and actually, because you have the whole system around it. Because in compost, you don't only have the microbes in there. Of course, there is soluble nutrients in there. There's a lot of more organic matter. So there's hormones in there. There's a plethora of fantastic stuff in there. So if you have a compost slurry, actually, it really aids the germination of a lot of seeds and growth of plants. But it's expensive, right? So that's why we only make an extract, take the rest of the material, put that back into the compost. It gets re-inoculated, right? It's difficult to make huge amounts of compost. That's why we're not always doing slurries, because then that's on the field. You know, that just depends on what your cash crop is all about, how much you want to invest.
So
Tanya,
when,
can I just ask a quick question, Tanya, about docks and thistles? So say you go to a dairy farm and they are really struggling with docks and thistles, and they've got a real problem in their head with them.
Yeah.
Do you say just leave them, don't just get used to them, and we're just going to try and overcome this within the soil?
No, actually, the thistles, they have a what do you call that root type again, Ian? Tapered root? No,
Taproot.
Taproot, yeah. So if a plant and comes up and it's a pioneer plant and has a taproot, it's indicating that you're having a severe compaction problem. So I say, thank you, thistle, for giving me the signal. Now, if it's just a little bit of thistle, a little bit every once in a while, you know, it's just not such a big deal. But if the field pretty much is dominated by thistle, then I know we have a compaction problem. And the easiest way to solve it is to really break it open, put the biology back in. It's the last time you break it open, and then put in a green cover and have above and beyond work together to solve the compaction. I've got a crazy story if you want to hear it.
Yeah.
The food forest that I've been working on, this is the second year. The first year I looked at the food forest, and it's on a grassland that has been neglected for years and years and years. I mean, years. So it was a completely clay soil. I couldn't even get in there with my penetrometer. It had sheep grazing on it every once in a while. But at this point in time, when I talked to the group who wanted to put the food forest in, I said, it is completely bacterial dominated. It's got root feeders everywhere. We need to do something. It might take more than one growing season. So we put in a green cover crop with lots of pen roots, tap roots, right? Tap roots, yeah.
Yep.
And at the end of February, I went to see how this was going, right? It had grown the whole year. So I look at the next year, and I already told the group, don't get excited. You know, we might not have done the job in this one season. But I went in there with my penetrometer where I could not, outside the fence, because I put a fence around it so nobody could walk over it. Outside the fence, I can't push in my penetrometer. I was hanging on it with my full 70 kilos. I couldn't get it in. So I thought, okay, now I go in there and let's see what the compaction went through. And I was, zoom, I went through it. I almost knocked out my back. That's how fast I went through it. I didn't expect it. So then I thought, okay, this looks good. I'll take some soil samples. And I went back home. Again, didn't feel very confident. It's at the beginning of the season. How much fungi am I going to see? And I was astounded. I came up with a F-to-B ratio of 3.0 with loads of amoebas in every field of view. I was just knocked over. So I gave the green light. You guys, you can put in the food forest. It's fine. So now they put in the food forest in 3 weekends. And today I went back to the field. And I took another sample because everything's been topsy-turvy, right? Everything's been dug up, not everything, but where they put the trees, it's dug up, and they walked around it in order to put in the trees. So now in the top 10 centimeters, I have lost a lot of fungi, but deeper in there, I'm sure they're still there. And I'm just going to wait until that food forest is going to start to grow. But I was just so blown away that such highly compacted clay soil that I couldn't get in, still can't get in outside that field,
Yeah.
It's cleared up in one growing season. I'm blown away.
Can I just, so you mentioned sort of the green cover, what sort of plants would be in it?
I have to look up the mix, but we really thought about the mix that we needed to have, because we needed some tapered roots, tap roots. I'm sorry, I keep forgetting that word. So it can penetrate that soil. And by the way, I couldn't even put a rototiller in it because I wanted to open up the clay to have the seeds that I was going to put in. I thought they would have a better time if I opened it up a little. I couldn't even get the rototiller in. Then I decided, okay, let's bring in a plow, but the plow person couldn't get through the gate. So that was the end of it. So then the only thing I could do is broadcast the seed, put a lot of vermicompost over it, and sprayed it twice with extract, thinking that's all I have right now. That's the best I have. But it all came up, it started to flower and it started to do its work. And I'm still flabbergasted. I can send you the mix, but I have to look it up in my graveyard.
Thank you.
Yeah.
On that, I don't know whether Jim Beery's on the call, but we're doing a little bit of work with Jim, and we've got baseline data from when before he moved into his new place, and the fields which he's put into a winter cover crop, and these are similar, probably going to be similar species to what Tanja talked about. A lot of them are the brassicas, the really deep-rooted radishes, and the idea, like when we did the baseline measures, there was a plow pan right across that field, and biology-wise, there was little in there, and water infiltration. Well, you could go away and have a cup of coffee and come back. It wasn't going in. So the plan, like Jim's sort of go-to, was this winter crop that he's used that winter stock on, really deep roots, and then it's going to be sort of gone into a herbal lay. So we'll hopefully in the next, depends how much frost and snow we get coming up, but where we don't want to be measuring microbes unless the soil's warmed up a little bit, but we can go in and see, we can measure the difference with the penetrometer and the biology, and it'd be really interesting to follow. And the other interesting part of that is that if we go back to that succession diagram, that Mother Nature would go weeds, then into brassicas before she moved on into grasses. So we're kind of working with Mother Nature. We're not doing a jump from ground zero straight up to grasses. That the brassicas he's had in there will have driven the microbiome that they want, and then that in theory should be a smaller step to go from that to grasses. But maybe we can do an update on that when we get other measurements and see how it goes.
Ian, is this a little Arcella hiding here?
Oh, are we? Just change the. Sorry, if we just change. I don't know how much time we got. The screen shared. Where are we? PowerPoint. Yeah. Is where are we showing the PowerPoint again?
You're showing the water tank, the brewer.
Cool. So if we just quickly go through these, Olivia and my mate Jim are possibly the world's fastest engineers. So the idea of this is on the right hand side, we've got a 1200 litre per minute trash pump that can handle big solids. And what's happening there, we're pumping out from the bottom of the tank into the trash pump, and then the output from that is split. So what we've actually done, where the hole is at the top, you would use as a priming hole, just on that little black line. That goes to that's our output for spraying. And then a lot of, so proportion wise, I'm getting, I guess, 3/4 of what we're doing is actually going back into the tank, and allows us to create this vortex. I've gone to the next video. And this vortex, stop that, then allows us, we can put solids in there. So I think Steve in New Zealand said he's at times gone 50% compost, 50% water. expensive in terms of compost, but you know, I'm thinking we depends on the biology, but we could maybe put 50, 100 kilos compost to 1,200 liters of water. But importantly, we can put seeds in there. So we're going to have a go at introducing and establishing herbal layers by banging the herbal seeds in there. But we can put our humics in there. We can put minerals in there if we need. We can put fish hydroslates, and this vortex keeps it in suspension. And then the last video, and I appreciate there's an irony in this, in a shed that's got a load of nitrogen at one side, but no. Are we blocking, can you see that, or are we blocking it?
No, we can see it.
So yeah. So that's where we are. It needs, I think, probably 20 minutes. We've got a leak somewhere, just sorting that out, and then we'll be calibrating it up, and we should be able to put a range of anything as low as 50 litres per hectare up to 200 or 300 litres, and it just gives us, you know, so many options in terms of what we can do.
So the plan, Ian, is that this sprayer will maybe be at carbon calling?
Yeah, so on the, I think the Sunday, did we say, Nick, that
well, probably
we'll hopefully have it along there, and maybe bring some of our other kit. If we just, where's the go backwards. So we've got this Bacchus compost turner, which this was the very first pile we did last year, and then unfortunately I bought a bit of a dog of a machine from Poland, and it took the rest of the year to sort of get it repaired and going. But there's been quite a lot of conversation within the soil food web group about windrow turner designs, and I chatted quite at length to a couple out in California. Again, worth looking up, Catalyst Microbe, they're called. So Keisha and Casey are so knowledgeable and been producing really, truly amazing compost, haven't they, Tanya?
Yeah, they have.
And what they pointed out with the Bacchus design is the teeth in the middle. So it's what happens with our compost piles. We have a hot center and a cold outside. So the teeth design, as it goes through, chucks the hot inside out to the outside, and then brings the cold outside into the middle. A lot of compost turners, the teeth are all pointing the same way and just chuck it up in the air. So you might get a bit of your hot center land in the hot center again, or some of it never end up in the middle. So for the kind of compost that we're looking at, where it's really important to get this diversity and quality of compost. We're not looking at waste reduction. This is about producing these really good quality compost that we can use as inoculums. Then this machine, yeah, we were excited to sort of get going with that, and can really upscale what we're doing. It's probably not going to be that good if you've got 500-600 ton of farmyard manure. It's just simply that you're going to need lots and lots of windrows. The height of that's just over 4 foot, and I think is it 6 or 7 foot wide. So it's not massive windrows. But yeah, it's we hopefully, if we can get geared up, we'll bring that along, Nick, and demo that. And the other one, if there's time, is the sort of, can we go back onto the no, stay on that, just go to the previous slide, please. Is the thermal method of doing these small boutique piles. That's sort of Elaine Ingham's type teaching. And is that the right? So another speeded up. So what we do, see the trugs in the background, they're 40-litre trugs, and we're starting with 30 of those. So they're 1,200 litre piles, and really precise recipe that 60% browns, 30% greens, 10% high nitrogen, the party food, as Elaine Ingham calls it, and making sure moisture's right. So as this is a starting point, we mix it in 2 goes, and we're checking that moisture's 50%, mixing everything together, and then it's so we've already put half in, and then the second half will go into these. That's on a pallet, a 4-foot pallet, and just with some fairly narrow gauge mesh around it. And work-wise, probably takes sort of 2 hours, maybe half an hour getting the ingredients and pre-soaking them, an hour and a half building it. And the key to this is avoiding recreational turning, that we want to turn this the minimum amount of times possible. So we know we've got a hot center, and we'll turn it 2 or 3 times. And each time we turn, the hot center will be, have we got another, where is it? Yeah, so these are in American numbers, but we've got that hot center and cold outside. So on turn 1, we will move the hot center into the bottom of our next pile, and then what was on the cold top will go in the middle, and at the bottom on the top. And if we repeat that 3 times, well, even 2 times, it means that every part of that compost has been in the center for the desired sort of period of time. Ideally, we're looking at, as Tanja calls it, the Goldilocks numbers of 55 to 65 degrees. And that would be 3 days at that temperature. If we get a little bit above it, we drop down to 48 hours. And if it's 70 degrees, 24 hours. 70 is, we're borderline there. It's so easy to go from 70 to 75 to 80. And at that point, your piles simply can't suck enough oxygen in, and our beneficial microbes start to die. But these turns, if looking at the first one, it may take sort of 4 or 5 days to come up to temperature, and then you've got another 12 days. So within sort of 20 days, 15 to 20 days, that's your turning done. And at that point, we want it to cool down, and the mesophilic, the cold loving microbes, and in particular the fungi, that's the point they'll start to proliferate. And then we're not going to go back in and turn that again. We'll look after it, we'll keep the water levels right, and we may well give it a fungal feed of a fish hydroslate or something just to boost the fungi. But with these piles, we can get I'm certainly getting piles now that have been, the 1-to-1 fungi to bacteria, and we've had piles that 2, 3, 4-to-1, but also getting a real diversity of nematodes and protozoa in there. And in this process, it might take 2 or 3 months for it to cool down, but as opposed to a Johnson Seuss static pile, which a lot less work, but you're looking at sort of 12 months before that's ready. We can start and get inoculums a lot quicker with this in sort of 3, 4, 5 months. And if we're only using, that sprayer we had before, if we were going 100 litres per hectare, 12 hectares, we might only use, 50 to 100 kilos of compost. These piles, by the time they finish, could be 300 or 400 kilos. So there's, we can do a lot of ground just with a small pile.
It's another
So
world, isn't it?
Nick taking pictures.
I know. Well, this is right. Yeah, this is like Renault's favorite thing, but he'll have to listen to it on catch up. But so Tanja mentioned in the beginning about trying to get dairy farms, working with dairy farms to be regenerative. So the bit that I may be struggling with is we do the compost stuff, and that's amazing. But you're a large dairy farm and you're producing loads and loads of slurry. Yeah. And surely when they put that slurry on, it's going to wreck everything.
Absolutely. And we're hopefully we put into the FIPL, the Farming in Protected Landscape people who've got the grants available in the national parks and the AONBs, asking if they would fund a trial. It was a little bit out of the list of things they were told they could allow, but they've gone to DEFRA, and DEFRA have approved that. And hopefully later this year, we're going to be doing trial work at, what can we do to treat these slurries so that we're not losing nutrients, they're not being gassed off, they're not being washed out into rivers, and we're not killing worms, we're not killing microbes, and we're making something that's beneficial without doing a lot of damage. And then things we're going to be trialing there range from the anaerobic products, the effective microbes, through to looking at humics, different humic products that'll lock up and chelate the soluble nitrates and other nutrients that when we're applying them out there, we're getting ammonia toxicity that's killing organisms, but it's big risk of runoff into rivers and things. So long list of things we're going to be looking at, and then there's different microbial products. So there's things that can be done, but it's it's not going to be straightforward. And the area where we live, it's in this sort of ideal world, you'd say, right, well, let's change our farming system so we're not producing slurry, but the price straw is to buy around here, it's just non-starter, absolute non-starter. And then there's a lot of issues with management of straw bedding and out wintering with dairy cows in this kind of area isn't an option. So it's got to be a real world solution. And what can we do about slurry? But there's a lot of products out there, and the thing we're keen to try is a combination. So the commercial companies are coming along and touting their microbial product, but they're not suggesting adding it with a humic or a biochar or some of these other things, which we're going to trial and hopefully get some ideas that we can move on and help, because there's absolutely no point us putting out really good biology and all the things to look after soil if we then slather it in slurry that's killing all those good microbes.
What's interesting is because between fungi, protozoa, bacteria, all found in rumens, so there is an inoculation coming out of those animals of those microorganisms that have flowed out of the rumen. So it's of value, isn't it? It's just how you transform it into something that isn't.
Absolutely, and to a certain extent, what's happening in the rumen is an anaerobic process, but the good anaerobes, and a lot of what happens in soil is aerobic processes, but there's also a lot of anaerobic processes, which is why these lines of looking at Bokashi treatments of farmyard manure and different effective microbes, there's a value in that. And that's how grasslands have evolved, isn't it? That it was a symbiotic relationship between a herbivore and a grass plant. The two have evolved together, and the interaction of the two. The real key to that, Liz, is where it comes directly out of a cow, it's straight onto the ground, and in theory we've got loads of dung beetles, we've got lots of worms and lots of things that are going to overnight pull that down into our soil, and it's going to be made use of pretty quickly. Where we've kind of gone wrong with farming is massive manure heaps and slurry tanks, because you suddenly end up with a really nasty, potentially nasty anaerobic process where the good anaerobes are outcompeted. And once we start smelling these things that we've called a good country smell, your eggy smells, your sulfurs and so on, and ammonias, they're not a good country smell, and they're only something that's sort of been around, maybe since the 60s and onwards. That, like, chatting to my dad, like he can remember as a boy that, all right, they didn't have many cows, but the muck was never stored. It went out straight into fields or tiny little piles that were there later spread, and you didn't get those nasty anaerobic processes happening. So it's but we are where we are, that we can't go out daily as if you've got a few hundred dairy cows, we can't be going out daily spreading that to replicate nature. Beef cattle, it's easier, that we're sort of replicating particularly with grazing management, what would have happened with herbivores centuries ago. But dairy cows, it's a bigger challenge.
But also Tanya, in terms of whether Tanya just wants to mention, do you mean we've got a few minutes left in terms of her experience with dairy farms in the Netherlands?
Right. Yeah, so dairy farms in the Netherlands are heavily regulated. So of all of the land that they have, 80% has to be dedicated to grassland, and 20% of the land can be dedicated to maize, which is cow maize, not cob of the corn, right? Yeah. And so we put our, the biggest gain actually is to get the grasslands to be weaned off the fertilizers. That's the biggest part. That's 80%, right? So we turn the grass around first, and then at the same time, we start to work on the maize fields by inoculating them already, but we continue with the fertilizer program because the costs of having to buy a lot of maize, protein food for your cows is also going to be costly. So it's doing it at the same time, but we get the grass first because that's 80%, right, of what they produce, build. Am I explaining it?
Yeah.
I think James Robinson asked a question in the chat, Liz. I've lost it now. About, where is it? Will you be ploughing the winter crop in before reseeding the herbal ley?
It's with Jim Beery. No, I think the plan is a light disc. I don't think there's a lot of stubble left, but I don't know whether is Jim on the call?
No, I don't think he is.
But yeah, I don't think there's a massive amount of residue left, and the idea is just a light disc.
And there's also a question, I think Tanya's just answering it now, but is there any good textbooks on compost production on farm?
Some of the early work that's really worth reading is Salbert Howard, who died around the Second World War, and he was sort of the godfather of a lot of composting methods, and it's his book, whilst it's fairly heavy going in the middle, the messages at the start and end of that are pretty mind-blowing, because he highlights all the problems that were starting in the late 1800s, 1900s, and suggests ways around it by using compost. Sadly, Second World War came along, and his message was lost, but yeah, him and Lady Evelyn Balfour would have been some of the early thinkers on it. The Rodale Institute have got a good book on different composting methods.
And so, just going back to carbon calling, if what would you suggest people bring in their little doggy bag? What just give us some examples.
Yeah, a really good point is if it's been a reasonable level of moisture, then in theory we should be seeing the microbes we need to look at in the top 3 inches, and try and take them from as close to a plant root as possible. If it's been slightly drier, maybe go a little bit deeper down. And really importantly is in that doggy bag, those microbes need air. We've got to keep them alive. So take the sample as late as you can, but when you seal your bag, seal it with lots of air in there. Because if you squeeze all the air out, by the time it comes to us to have a look at, there's a lot of things going to have died and changed in there. So lots of air in there. And we don't need masses. We've got 100 grams, 200 grams, not masses. We just need a little bit. So soil sample, and bring something along of what you maybe hope is your best ground, and what you think is the worst. And similar principle, if you've got some compost, bring those along, but just make sure that there's air in that, and don't go and put them in so it's June, so don't go and put them in the fridge overnight. Those microbes have sort of got used to a warmer soil temperature. Just if you're taking it the day before, leave the bag somewhere at a similar temperature, maybe in an outside shed to what the microbes would have been in the soil.
We had a chat with Ian in the day. I've got a field with fairy rings, lots and lots of fairy rings, and the sheep absolutely nail the grass around those fairy rings. So I'm going to bring some from the fairy rings and some from nearby.
Nice.
Or post it, actually, I might do. But anyway, just very quickly. So James Robinson has asked, could you make a small amount of high quality compost and then be mixed with a larger amount of farmyard manure before spreading? Or does all of it then have to go through, and you're basically using that compost as an inoculant to then fully compost the farmyard manure?
It's something that we do is use a good compost to inoculate other composts, and then definitely why not try it with the farmyard manure? I don't know, Tanya, have you any?
Yeah, well, the manure is, because all of our feces, the last part of it becoming feces is an anaerobic process. Up until that, in the small intestines, it's an aerobic process, so it's a lot of oxygen. But the last part is to withdraw as much water out of the feces in any mammal, right? And then the feces is anaerobic. So it usually has anything that we as humans or animals can't use. Pathogens, parasites, anything we have to get rid of from our bodies. So that's why it's actually pretty much important to do something before you apply it to your land. Because why would you introduce a parasite or an anaerobic? It's really hostile to life, really. So if we can do anything, and I do that with my dairy farmers, I have these big IBC crates. Is that a familiar word? Yeah. So those IBC crates, you can either take the plastic out and line it with landscape cloth, or you leave the plastic in, but then you take the top off, right? And you can fill that with a recipe of 10%. There are lots of recipes, but if you at least mixed 50% of your dairy slurry with 50% of wood chips and leave it there for a while until it's turned around, because then it will turn around and become more composty. This will take like 8 or 9 months. If you have any worm composting, compost worms, and you can add that in, it will go quicker. It will shave off months of maturity. So you can shave off to 6 months, then that's really, really great stuff to put on your land. You just have to get into the habit of doing something with the slurry in order to improve it just a little bit, at least, and then put it onto your field.
I guess the problem with that is, where we've got 500,000 litres, 1 million litres, it's not such an easy thing to scale up. But maybe James's question was, I think, probably more farmyard manure with straw, I'm guessing, is it? So
yeah.
So whether that's probably the easiest route in terms of handling a lot of volume is looking at the bokashi treatments. The problem we've got with if you've got hundreds of tons of farmyard manure, and to get a good balance with those brown woody ingredients, so the one that's on screen there with 60% woody material, it's going to need hundreds of tons of wood chip or straw added to it, and it's just not a sensible suggestion. So I think trying to work with what we've got to improve it into, at least make it into something that's soil friendly, whether that's an aerobic approach and you're turning it, and maybe looking at adding carbon in the forms of humics or biochars, or you go down the anaerobic route of the bokashi route, which is another. Bokashi is not something I know a lot about, and we're hoping to get some trials set up at gyms this year as well, comparing an aerobic process where we'll add in humics and biochars and similar things with an aerobic turn process and the bokashi, again do sort of before and after soil analysis. But yeah, it's a difficult one. It's purely because of the quantities. I think if James has some really good compost, he'd probably be better at looking at that as an aside to use on as an inoculum on his soil, and then looking at the large quantities of FYM and what can be done to make that soil friendly.
Thank you. Just over time, but I think I have a feeling we've only just scratched the surface of this topic. I feel like I'm now even more confused than when I started. So that's always a good sign.
Sorry.
No, just as you say, it's incredible. Yeah, it's very clear that we don't know. We know a very small proportion of what's going on in the soil. But thank you very much. Nick, did you want to add anything?
No, I don't. I just think I do really love all this, and I think that microscope stuff is really amazing. I'm hoping we're going to be able to twist Tanya's arm to come here in June, and it is definitely, it's the future, I think.
Yeah.
Did you get that, Tanya? A little holiday at Carbon Calling in June?
Cumbria is lovely.
I can swim.
Absolutely. We'll pick you up. We'll put you up.
I'm quite near the East Coast. It'll be fine. But no, thank you very much. Thanks, Ian. Thanks, Tanya. Thanks, everybody, for the questions. Yeah, interesting. Really, really interesting. So yes, looking forward to the session.
And Ian, make sure your spray is in fine fettle.
I
No
will.
pressure.
We will.
No,
we will. It should be fun, and hopefully, if there is questions that haven't been answered today, then maybe we can pick up when we've got a little bit more time at Carbon Calling, or feel free to ping me an email or a social media message. Or
I was just going to say, Ian, how do people contact you on Twitter?
Yeah, so Twitter and Instagram. I think with the links on the...
Yeah, I sent them out on the invite today.
Or was my email address on there?
What I'll do is I'll send another link out to the people who attended with the contact details.
Nice. I'll because there's quite a few slides we never looked at. I can package that up and send it across if that's of use to send out to people.
Okay, yeah, if anybody's interested, I can, if you send it to me, then I can do it. I'll do it on request thing. That's fine.
Nice.
But no, thank you very much, everybody.
Thank
good
you.
evening. Was hoping to finish before I sneezed. I apologize for that. I could feel it brewing. It's my microbiome that's mainly sheep at the moment. Just trying to get out of my body. Anyway, on that note, good night. Bye, everybody.
Bye.
Good night. Thank you so much.
Thank you.
Thank you.
Bye.